The Cell Cycle by Caitlin,
Natanya,
Kurt,
and Daniela.
In a dividing cell, the mitotic phase alternates with interphase, which is a period of growth. The first part of interphase, G1, is followed by the S phase, when the chromosomes duplicate. G2 is the last part of interphase. In the M phase, mitosis distributes the daughter chromosomes to the daughter nuclei, and cytokinesis divides the cytoplasm, producing two daughter cells. G1 of interphase is the first gap where the cell begins to grow. S is for DNA synthesis, in which the cell continues to grow and copies its chromosomes. G2 is the second gap where the cell continues to grow in preparation for cell division. M is for mitotic phase, where the mitosis and cytokinesis of the cell occurs. Within the mitotic phase includes 5 of the following phases: prophase, prometaphase, metaphase, anaphase, and telophase with cytokinesis.
In prophase, the cell begins to break down some structures and builds others up, which prepares the cell for division for the chromosomes. This leads to prometaphase, where the mitotic spindles begins to capture and organize the chromosomes. The nuclear envelopes breaks down, releasing the chromosomes. Next, in metaphase, the spindles capture all the chromosomes and lines them up at the middle of the cell, preparing for division. Next is anaphase, where the sister chromatids separate from each other and are pulled towards the opposite ends of the cell. Next is telophase and cytokinesis. In cytokinesis, the cell is nearly finished dividing, and it starts to reestablish its normal structure as cytokinesis. The division of the cell contents takes place. Finally, the cell undergoes the G0 phase, which is a non-dividing stage where a cell exits the cycle at a checkpoint until it receives a go-ahead signal at the G1 checkpoint.
MPF activity. Kinases drive the cell cycle, but they must be activated by the attachment of a cyclin. Cyclin-dependent kinases are enzymes that phosphorylate specific target proteins. The attached phosphate group acts like a switch, making the target protein more or less active. When a cyclin attaches to a cyclin-dependent kinase, or CDK, it activates the CDK as a kinase, but it also directs the CDK to a specific set of target proteins, ones appropriate to the cell cycle period controlled by the cyclin.
Checkpoints in cancer cells. Cancer cells show a loss of the normal cell cycle controls and may divide without being checked in the 3 checkpoints. It can result in malignant or benign tumors. The 3 checkpoints are located in the G1, G2, and M phases. In G1, if a cell receives the go-ahead signal at this checkpoint, the cell cycle will continue. It is regulated by the cyclin CDK complexes. In G2, when sufficient MPK accumulate, the G2 checkpoint is passed and mitosis is promoted. In the M phase, the kinetochores must all be attached to the spindle fibers during metaphase. This will activate an enzyme which allows the sister chromatids to separate.